Hello everyone, welcome again to Toppr.com. So this is our first live video lecture session on biology. I am Pushpendu. I am a subject matter expert in Toppr.com. So I am sure you must have seen me before in our different webinars that we have conducted, right? So based on your responses, we got actually very good responses from you. Thanks a lot for that. And because of your responses, we have started a new session, that is live classes. Okay, so we have been conducting live classes in physics, chemistry, mathematics, and today is the first session for biology. And in this session we are going to talk about a very basic topic, very interesting topic of class 11th biology, which is cell cycle and cell division. Okay, so I'm sure you have heard about this topic, right? Cell cycle and cell division. So before starting this, let's talk about the word cell. Okay, so what is cell? Correct? We know the cell, it is the basic structural and functional unit of life, right? So it will be the basic structural and functional unit. Okay, so now what we need to know, these are prior knowledge that you must have so that we can understand this session, is we should know about the cell structure, we should know about the nucleus. Okay, nucleus. So what is nucleus? So it is a dense matter which is present inside the cell. And what is present inside the nucleus? That is the genetic material. Okay, we have the genetic material. Now, what is this genetic material? If I ask you what is this genetic material in case of human beings, this genetic material for human beings is DNA, right? And this DNA, I'm sure you must know about it, that it remains in a chromatin form. Okay, chromatin. So what is chromatin? So if I draw DNA as this length of DNA is about 2.2 meters, right? And actually inside the nucleus, this DNA stays as chromatin network in this form, okay, as a network inside the nucleus. Now we'll see different forms that is forming during the process of cell division. Now in this process of cell division, a certain structure forms, and that is called as, I'm sure you must have heard or you must have seen this structure. It looks like a butterfly, but it's not a butterfly. It looks like a butterfly, and this is called as a chromosome. Okay, this is a chromosome. What is chromosome? Chromosome is nothing but a condensed form of this chromatin network that we have drawn here, right? It is a condensed form. There is a high level of supercoiling. बहुत ज़्यादा supercoiling करके ये जो structure है, this structure is attained. So which is known as the chromosomes. And how many chromosomes do we have? The human cells, we have how many? 22 plus 1 and pairs. Okay, we must know about this. So we have 22. Why have I written it as 22 and not 23? So it is a form of representation, but you should know 22 means these are the autosomes, right? And this is the sex chromosome, which determines the sex of an individual, that is if it's male or a female. So we have in total 23 multiplied by 2, that is 46 chromosomes, correct? If you take any cell other than the gametes, there is 46 chromosomes in case of human beings. Now this number actually varies in different organisms. अलग-अलग organism में अलग-अलग number of chromosomes होते हैं, correct? So now, why, the first question, the very basic question is, why cell division? Why the cell needs to divide? So you think that there is a cell, now it has to grow, it has to increase its number, correct? So how will it increase its number? It can increase its number by the process of cell division, correct? So through this process of cell division only, this number can be increased. And this cell division is the reason why I'm growing, right? During when I was a kid, my size of my body was quite small, काफी छोटा था मैं, right? उसके बाद, due to the process of cell division, what happened? I grew up, I have grown in size, I have grown in my height, एंड ऑल दिस हैपेंड, ये सब पॉसिबल हुआ क्योंकि मेरे सेल्स बॉडी के अंदर वो डिवाइड किए. नाउ इस प्रोसेस ऑफ सेल डिवीजन को जानना बहुत इंपॉर्टेंट है. सो whenever you study something, you should know why you are studying this, correct? We should know why are you studying cell division. You should ask this question. So if we study cell division, we can solve many types of diseases, correct? So we are able to solve many types of diseases if we know about the process of cell division. Okay, so cell division is the process in which new organisms are formed. Now you think, if there is a flaw or there is a defect during this process of cell division, then the organism that will be formed may be defected, or in fact an organism may not be formed. So we find many types of defects that is happening in case of babies that are taking birth, and this actually happens because of faulty cell division, specifically in our germ cells, that is the reproductive cells. So let us see why this is important and how this process is taking place. So this process of cell cycle and cell division, as we have talked about it, it is very important. Let's see what are the different stages a cell cycle or a cell division has. So first we'll be talking about the cell cycle. So let's talk about the cell cycle. Okay. So this word cycle, right? I'll show you why we call it as a cycle. It's actually represented by a wheel of a cycle. Okay, it's a circle and it's a cyclic process. That's the reason it is known as a cycle. Now I can divide this into several stages. Let me divide it, and this diagram, please take a note on this diagram, because very often during your board examinations, your class examinations, these diagrams are being asked. Often these diagrams are asked. So we'll see the different stages. Basically this cell cycle process is divided into two phases. Okay, remember it. The first phase is known as the interphase. Okay, and the second is known as the M phase. Now let's see what are these two phases. So the first phase, interphase, how much? So let me mark it. This is the part which is actually represented by the interphase, this much. Okay, so this is, let me represent it as I, this is interphase, and if I represent the next one, which is this part, this is the M phase, correct? You got it? So M phase and this interphase. So let's talk about these phases and understand what is a cell performing during these phases. So which phase are we supposed to talk about first? Let's talk about interphase. Interphase, this is the first phase. Okay, this is the preliminary phase. जब एक सेल डिवाइड कर रहा है, राइट, उनको फर्स्ट इंटरफेस करना पड़ेगा. इनफैक्ट वेरी इंटरेस्टिंगली, पहले साइंटिस्ट सोचते थे, साइंटिस्ट यूज्ड टू थिंक दैट दिस इंटरफेस इज this phase is a resting phase. Okay, resting phase. Now why resting phase? Scientists thought that the cell is doing nothing in this phase and the cell is actually resting in this phase. So what happens during this phase? The cell is resting. अब क्या होता है, एक्चुअली बाद में ये चीज गलत प्रूव हुआ कि ये रेस्टिंग फेज नहीं है, क्योंकि इन दिस फेज द सेल इज वेरी हाईली एक्टिव. बहुत ज्यादा एक्टिव है. पर साइंटिस्ट पहले सोचते थे कि वो एक्टिव नहीं है इस फेज में. अब देखते हैं क्यों हाईली एक्टिव कहते हैं. इस इंटरफेज को अगर हम देखते हैं, ये जो फेज है, पर इसके अंदर मैंने 3 कंपार्टमेंट्स बनाया है, राइट. नाउ सी, दिस फर्स्ट फेज इज नोन एज द S1, इट इज नोन एज द G1. ओके, सो आई हैव अ क्वेश्चन. दिस इज हेल्पफुल फॉर नीट. Is this topic helpful for NEET? Of course, this topic is very much helpful for NEET, because this is one of the basic requirements which you should know. Then only you will understand various problems, specifically when you move on to your 12th. Okay. There you will find genetics, there you have to solve problems, there you have to understand a very famous scientist's name, that is Mendel. Okay, he's called the father of genetics. उनका काम समझने के लिए you have to understand this process of cell cycle and cell division, right? So all the best for the NEET, because you have asked this question and this topic is absolutely important for NEET, and definitely you are getting questions on NEET from this topic. So let's continue. We have this first phase which is known as a G1, right? Now what's the next phase? Next phase is called S phase, and we have the third phase which is called G2 phase, right? So G1, S and the G2 phases. So let's talk about these phases in detail. What a cell is actually performing in these phases. So which one will talk first? G1. G1 is known as gap 1 phase. Okay, gap 1 phase. Now you understand that this phase, interphase, is totally a phase in which it is called a preparatory phase. That means एक cell अपने आप को prepare कर रहे हैं, right? Cell को क्या करना है? Divide करना है. Cell wants to divide. So before dividing, the cell needs to prepare, right? So this is the phase. Interphase is the phase where the cell is preparing itself. So let's see how the cell prepares, right? So this is the first preparatory phase, that is gap 1. G1 is the first preparatory phase. Now, what are the things that a cell is preparing during this phase? Let's see. So in this G1 phase, a cell is making proteins. A very important molecule is being synthesized at this stage, which is ATP. Okay, what is ATP? ATP is the energy molecule, right? So we perform all the different activities that we are doing every day, every moment. That is possible because of this molecule, which is ATP, correct? So this is the energy molecule. This energy molecule is constantly generated during this gap 1 phase. So what are the preparatory phases? So here in the gap 1 phase, proteins are being made, ATP is generated, enzymes. Later on, the stages, various stages, we are going to talk about those. All activities of those stages are being carried out by these proteinaceous structures, which is called as enzymes. So enzymes perform करते different metabolic activities or different जो work हो रहा है एक cell के अंदर, those are performed by the enzymes, correct? So these are some of the important molecules which are being made in gap 1 phase. So can we talk about the next phase, S phase? So S phase is known as the synthesis phase. Synthesis phase. Now, can you guess कि ये नाम क्यों दिया हुआ है? Why synthesis? Because here DNA is replicated or duplicated. Okay, replicated or copy of the existing DNA is being made. So that's the reason it is known as the synthesis phase. Why? Because a copy of the DNA is being synthesized. So basically, this is a very important question which is being asked in examinations, is which is the phase in which DNA is copied? Simple answer is the S phase of the interphase, correct? So now, is a cell performing any other activities in this phase, S phase? Yes, the cell is making histone proteins. Histone proteins. So do you know what these histone proteins are? What are the functions of these histone proteins? These histone proteins help in the coiling of the DNA, right? So you know the DNA, if I draw, it's like a strand, okay? It's as a network we have seen. But in the form of chromosome, it stays in this form. So how come the transition from here to here? This transition happens because this histone proteins actually helps in coiling the DNA. So because of coiling, बहुत ज़्यादा coiling होता है, जिसके वजह से ये chromosome structure attain कर पाते हैं, right? So chromosome structure forms because of excessive amount of coiling brought about by the histone proteins, and it is being asked where the histone proteins, in which stage of cell cycle the histone proteins are being synthesized. It is the S phase. So we have covered G1, S. So what is left? we are left with the G2 phase. Let's see what a cell is doing in G2 phase. So the proteins, which are very important, you see, because we have talked about that the enzymes carry out different actions. So these actions, for these actions, protein synthesis is very essential. So proteins are being synthesized. Okay, various ribosomes. Now, what are ribosomes? Ribosomes are the areas where proteins are synthesized. Okay, so ribosomes also need to be synthesized. In addition to that, various cell organelles. Various cell organelles are being synthesized in this G2 phase. Correct? Because you understand, the cell has to undergo division. That means in the two cells, all the components of the cell must exist. So cellular organelles has to be multiplied or duplicated. So we have talked about the three stages: G1 where proteins and ATP enzymes are made, S phase where synthesis of the DNA is being made, histone proteins बनाया जा रहा है stage पे, and G2 जहाँ पे proteins, ribosomes, cell organelles कॉपी किया जा रहा है, right? प्रोटीन्स बन रहे हैं, राइबोसोम्स भी बन रहे हैं. So हमने बात किया इंटरफेज. अब इंटरफेज के बाद कौन सा फेज आएगा? क्योंकि जैसे हम डिस्कस कर रहे हैं, द वे वी आर डिस्कसिंग लाइक G1, S, G2, इसी तरीके से एक सेल परफॉर्म करता है अपना एक्टिविटीज. सो G1 के बाद S आएगा, S के बाद G2, देन विल मूव इनटू द M फेज. ओके, सो एक सेल G2 के बाद पहुंचेंगे M फेज पे, विच इज द मेटाफेज. ओके, इट इज एक्चुअली द माइटोटिक फेज. इट इज नोन एज अ माइटोटिक फेज, एंड इंटरेस्टिंगली इसी फेज में एक सेल डिवाइड करते हैं. ओके, दिस इज द फेज इन विच द सेल डिवाइड्स. Correct? Now I'll tell you about a very interesting fact over here. Here at times, so once a cell goes to M phase, that means it has divided and formed two cells. Correct? After that, again when it needs to divide, it will again, those cells will again go to the G1 phase and then S phase, then G2 phase. But at times there is another phase which the cell moves is G0. It is known as G0 phase, or it is also called as the quiescent stage. Okay, G0 or G0, or this is also known as the quiescent stage. Now what is this quiescent stage? So okay, I think I've got a question about histone proteins, right? So if you did not understand about histone proteins, so I'll explain you once more. This is the DNA strand, correct? And I have said that in the cells, during the process of cell division, क्या होता है? सेल को अपने DNA इस तरीके से करना पड़ता है, right? It has to form the chromosomes, correct? Now, just imagine it is a straight structure. From this straight structure, how will this butterfly-like structure, which is a condensed, very short structure, this will form? This can form if the histone proteins are there, you know. So these histone proteins, I'll show you. These are proteins, okay? As you can see here, I am drawing, and the DNA is being coiled over it. You understand? Have you seen a rope which is coiled on a particular surface? You understand? A rope is very long. To make it short, we coil it over a particular surface. So that is the same thing histone proteins help. It helps in the coiling of the DNA. Right? I hope you have understood. So can we move on? So we were talking about the quiescent stage. This is an interesting stage where the cell, if it goes to this quiescent stage, then the cell do not divide further. This is a stage where the cell arrests its division. यहाँ पे सेल अगर पहुंच जाते तो सेल डिवाइड नहीं करते. सो क्वेश्चन ये कि क्विसेंट स्टेज पे जाने की जरूरत क्यों है? वाई? क्यों जरूरत है? क्योंकि नाउ इमेजिन मैं ग्रो हो रहा हूँ, राइट? आई एम ग्रोइंग. नाउ आई हैव रीच्ड अ स्टेज आफ्टर विच माई हाइट इज नॉट ग्रोइंग एनीमोर. My lungs, my heart, they are not growing anymore. Imagine a condition, अगर मेरे height, मेरे lungs, मेरे heart ये grow करते रहे, तो what will happen? You understand? I would have become, till now, I would have become with that height, right? Which is like you can consider the height of a coconut tree. But that does not happen. Why? Because my cells have undergone the G0 phase. It is a phase where the cell is temporarily, you should understand this, that the cell is not dying, it is not arresting permanently, but it is arresting temporarily. Okay, so let's see if we have got some questions. Okay, so I have got a very basic question. Okay, Edward, that what is chromosome? Okay, fine. So let's see. Chromomere. Okay, you are asking chromomere. Fine. Chromomere. So you understand this structure of this chromosome. Okay. Here, now these are actually two strands. Okay. If I separate them, it will become these strands. Correct. Now you understand why I have drawn like this. It is attached. Correct. So here these two are being joined together. And this center is also known as centromere, and each of them, they are known as the chromomere. Understand? So this portion where both of them are attached is centromere. Each side there are actually, you will learn in your higher classes, there is kinetochore which actually helps. It is proteins, kinetochores. There are proteins which help in this attachment, and these are chromomeres. Okay. Very constricted area. These are the very constricted areas. Correct. So again, let's move on to the G0 phase. It's very important. So you understand G0 phase is important so that the cell is temporarily arrested. They do not divide. Correct. Now this stage is so important because once, suppose I have a cut in my hand. So what will happen? My cells have arrested already. So but the interesting fact is that the cells will again revive and it will start dividing. So this is what I'm mentioning. It is a not permanent. It is a temporarily arrested stage. Correct. So now we have talked about the interphase. Let's move on to the next phase. So I will be changing the paper. So see this, we are talking about now this M phase. Correct. We have covered this interphase. Now we are going to talk about the M phase. So let's see what is happening in the M phase. So here we go. We are going to talk about the M phase. So cell cycle divided into two phases, interphase, M phase. Now M phase is again divided into two phases. What are they? Karyokinesis. Okay, karyokinesis, and the second one is known as cytokinesis. So I'll teach you how to understand what are the different names. Okay. And so while I'm teaching, so you should keep asking questions so that I can solve your doubts. Okay, because through this mode you may not understand some part, some questions may arise, so definitely ask, post it. Okay, I'll be answering your questions. Okay, so let's carry on. So M phase has two parts, two phases, that is karyokinesis and cytokinesis. Karyo or karyon, this word has come from the word nucleus. Kinesis means division. So what does this word mean? Karyokinesis. It means division of the nucleus. Nucleus के division को कहते हैं karyokinesis. Now I'm sure you can tell cytokinesis. What will it mean? Cyto, it is related to the word cytoplasm. So cytokinesis is basically division of cytoplasm. So we are going to divide the M phase into two separate phases, karyokinesis and cytokinesis. So which one should we start first? It is karyokinesis. Correct. So let's start it. Karyokinesis is basically division of nucleus. Now here I'll state you three important types. This division of nucleus can be of three types. What are they? Mitosis, mitosis and meiosis. So these are the three important types of karyokinesis. Okay, so first I'll talk about the mitosis, then meiosis will be a second one, and lastly we'll talk about mitosis. Correct. So let's start our discussion with mitosis. Mitosis, this is known as equational division. Now why equational division? Very interesting. So you'll understand this when we talk about mitosis. Correct. So let's start our discussion. So this is a very interesting chapter because आपको ये चैप्टर सिर्फ डायग्राम से समझ में आ सकता है, राइट? सिर्फ डायग्राम अगर आपको समझ में आए, इफ यू अंडरस्टैंड द डायग्राम्स, you are well versed with this chapter. Let's draw. So we'll draw a cell, and inside that we have nucleus. So from now I will represent the nucleus only. Okay, you take into consideration that I am showing the nucleus. So let's draw the nucleus. Here we have the nucleus, and inside the nucleus what we have? We have chromosomes or chromatin network. It's a chromatin network. So initially it is present as network. So here we have the first phase, which is known as the prophase. correct? So let's see if someone is asking any question. Yes, so I've got a question. Mitochondria is found in G2 phase. Yes, like I have said, many cell organelles, they are formed in the G2 phase. Yes, mitochondria is also formed in G2 phase, right? So the first phase, prophase, correct? Now in this phase, what happens? I've said that this is the area in which it is staying as chromatin network inside the nucleus. There is nuclear membrane, correct? Now let me draw the cell outside, then we can understand. So in this prophase, what happens? The cellular membrane, it dissolves. The cellular membrane, it's not the cellular membrane, it's a nuclear membrane which dissolves. Okay, so there is nuclear membrane. Nuclear membrane will dissolve. Now what else will happen? There is very high condensation of these chromosomes. The chromosomes will condense and it actually will form these chromatin, these chromosomes. This chromatin will condense to form the chromosomes, okay? Because now we are moving towards the cellular division. Also, we see the movement of the centrioles. So there are two centrioles. The centrioles, they start moving towards the poles. So this will come here and this will come here. So they start moving. They have not come, but they start moving. Also, we see the formation of spindle fibers. Okay. So let's revise what we have seen in prophase. In prophase, the chromatin network is present. It's condensed to form the chromosomes. उसके बाद क्या होता है? Centrioles जो है specifically animal cells में वो पहुंचते poles पे. They start moving towards the poles. The nuclear membrane, it dissolves, right? And we see the formation of spindle fibers. So we are done with the prophase. Now let's move on to the next phase. Next phase is called metaphase. And this is the phase actually which we can visualize under microscope. Under normal compound microscope we can see. So compound microscope पे अगर हम देखते हैं metaphase stage में, तब अच्छी तरीके से chromosomes दिखता है. Okay? So how it looks? This is the cell. Now will we draw nuclear membrane? No, because it is already dissolved. So we have the chromosome, correct? We have the chromosomes. So there should be, in animal cells, there should be like how many? 46 chromosomes, correct? These chromosomes, they are aligned at the center of the cell. So if this is the cell, the chromosomes will be aligned in this way. You see, chromosomes, they will be aligned at the center of the cell, and from here the centrioles, we have what? The spindle fibers. So these are the spindle fibers you can see. Very interestingly, there is polymerization and depolymerization, because of which they actually do constant pulling and pushing. Because of this pulling and pushing, what happens? This chromosome you are seeing over here, they are all aligned at the equatorial position. You see, this is the central position of the cell. So cell के बीच में वो align हो जाते हैं. So ये है characteristic feature of the metaphase. और very interestingly, हर एक पोल से जो spindle fiber आया है, that is attaching to the centromere of the opposite side. So from here one will attach here, from here one is attached, correct? So this is the stage. Can we move on to the next stage? So keep in mind about this stage. So let's move on to the next phase. Prophase, metaphase. Now let's talk about the third phase. What is that? Third one. It's called anaphase. So what was there? We had the cell and we had the chromosomes which were aligned at the center. Okay, so let me just check if you have any questions. Okay, very interesting. Then how will, if mitochondria is present in G2 phase, how will the energy be produced? So yes, good question. But the cell already has mitochondria, right? What I said is in the G2 phase mitochondria is duplicated, correct? Even the cell has DNA, correct? The cell has DNA, but in the synthesis phase the DNA is duplicated. Why? Because the new daughter chromosome, it has to have that mitochondria, has to have all the cellular organelles, has to have the DNA. So in this phase it is duplicated, okay? So very good question, very good thought. So can we move on? Anaphase, right? In metaphase we have seen the chromosomes have aligned at the equatorial position. Now what happens? Here we have the centromeres, correct? Now they pull apart these chromosomes and they are separated. You see, that means they were initially together, right? Together like this. So two centromeres, they had two spindle fibers as attached to the centromeres. Now they have pulled them apart, right? This is what has happened over here. This is marked as the anaphase. And please mind it, this is the cell, not the nuclear membrane. It is not the nucleus, it is happening inside the cell because nucleus is already, nuclear membrane has dissolved. So here this is the anaphase where what happens? The centromere, it divides. The centromere has divided, correct? And we have the sister chromatids. These are known as the sister chromatids. Together they were the chromosomes. These are the sister chromatids, and because of depolymerization, the structure, the length of these spindle, they starts decreasing because of depolymerization, okay? So it starts pulling it towards the poles, correct? Now let's move on to the fourth phase, which is telophase. So once again let me check if we can get some questions. Polymerization, polarization and depolarization, metaphase again. Okay, so right, I can show you the polymerization, depolymerization. I'll just show you here. So these spindles you are seeing, right, here I have just drawn with a single line, but actually these are not lines. There are certain microtubules. These are called microtubules, which is formed of tubulin proteins, and they are formed like this. Can you see here? I hope you can see. So they assemble together and they coil. Because of coiling, right, they coil like this. You understand, these proteins, they coil like this and form this structure. So this happens because of polymerization. Polymerization means it assembles. Now when it depolymerizes, it actually opens up. These come out. So what will happen? If these come out, the length will decrease, right? So it will result in the decreasing in length, and it will pull this sister chromatid towards the poles. Yes, you may ask a question. It is attached here, correct? Then if it depolymerizes from here, then what will happen? It will release the chromosome, but no, the depolymerization happens a bit ahead from this area, not absolutely the end, correct? So let's quickly move on because we have a lot to talk about now. So in telophase what happens? The reverse, telophase is actually called the reverse of prophase. So again I think we are getting good questions. Okay, so you are talking about RBCs, but in fact RBCs actually lose their nucleus and their mitochondria not in the initial stage. You know, RBCs, when they are formed from the bone marrow, they have the nucleus, but because when they mature they lose them. So you understand, and RBCs actually do not divide, correct? RBCs, they are formed from the bone marrow. Okay, so they have 120 days of lifetime. They do not divide to form new cells because you understand they do not have a nucleus. How will they divide? But in the preliminary stages they have their nucleus, correct? I hope I have cleared you out. So what happens in the telophase? So this is the cell. The nuclear membrane comes. So these chromosomes, these sister chromatids, they have assembled over here at the poles. Now there is formation of nuclear membrane. Nuclear membrane again starts forming. The spindle fibers, as we are seeing over here, they dissolve. The nuclear membrane forms. The centrioles, they also dissolve. Slowly and steadily they move ahead, and there is formation of what? The nuclear membrane. So you see, we have two nucleus inside the single cell. Now what should happen? Next, we have talked about the next phase after karyokinesis, which is cytokinesis. That means we'll talk about the cytokinesis. In animal cells, The cytokinesis, it happens in this way. I'll show you. So if this is a cell and if I have the nucleus over here, suppose I have the chromosomes, correct? Now what happens? There is a furrow formation. It's a furrow formation like this. Slowly and steadily this deepens and it joins. What will happen? It will now form two cells. It will just pinch out. It was like this, correct? Now it will slowly form this and will pinch out. Okay? So here we have the new cell, correct? And if you talk about plant cells, here if I draw this plant cell, here there is additional one thing, there is cell plate formation. Cell plate forms over here, okay? And then it actually, the cell plate joins these end areas, the cell walls, and it divides into two, correct? So this is the difference between animal and plant cell cytokinesis. So you understand we have got two cells. So let me check if we have some questions. Okay, so I'll just carry on with and entertain you questions after I talk about a bit more. Okay, so let's see. We have seen the two cells from plant cell and the animal cells we have seen. So just if we try to revise, recall what we have seen in this topic of mitosis. Here that this is equational division. That means you see both the cells, they have equal amount of DNA, equal characteristics of the DNA. That's the reason it is known as equational division. Okay. So remember this stage because we are going to talk about the next type of division where we will require this knowledge. So let's talk about now meiosis. Meiosis is basically two types: meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So these are the two types, correct? Let's talk about meiosis 1 first. In this meiosis 1 stage, what happens? There are four stages normally, that is prophase, metaphase, anaphase and telophase. Now here the prophase, let's talk about the prophase first. It has actually 5 different phases. Okay, first is known as leptotene. Here what happens? The chromosome which was present initially as chromatin network, it starts condensing and it forms the chromosomes. This is the characteristic feature of the leptotene, correct? Next is zygotene. Zygotene is the stage of prophase 1 of meiosis 1 in which bivalents form. What forms? Bivalent. Now what is bivalent? To understand bivalent, you should understand about homologous chromosomes. What is homologous chromosome? We have these pairs, right? We were talking about pairs of chromosomes. So from where these pairs come? One come from your mother, one come from your father. So one is maternal, one is paternal chromosome. So they form bivalents. Initially they were in a mixed up stage, mixed up form. Now during the zygotene, these homologous chromosomes, they start pairing up. That means if this is a chromosome, it will pair up with the next one only, the homologous, the similar type. If this is another type, that is chromosome 2, so it will pair up with chromosome 2 only, correct? So we have two different types of chromosomes, homologous chromosomes, they pair up. So this is called as bivalent formation. So question is often asked, कि bivalent कहाँ पे होता है? Where is bivalent formation happening? It is happening in zygotene, correct? Next, we have pachytene. Pachytene stage. This is the most important stage. Why? Because here a very interesting thing happens. That is recombination. I'll show you. So let's suppose this is one chromosome. This is the second chromosome. Let's suppose this is from father. This is from mother. Now there is recombination, right? There is some recombination and genetic exchange over here. Now this can result into the formation of a chromosome which may look like this, which may look somewhat like this. Okay, so you understand here. So here it has exchanged this pink part correct? You see, pink part has been exchanged. In fact, the other way around, same thing happens over here, because certain exchange happened. Here, certain blue part has been exchanged. So because of this sexually reproducing organisms show variations. Okay, you understand? So this was one gene. Now it has recombined and formed a new type, new form, new characteristic features can arise because of this recombination. Okay. So this stage of Pachytene where crossing over actually is happening, this is known as crossing over. This can be visualized in the next stage, which is known as diplotin. Diplotin, why it is visualized over here? Because this crossing over, this point where crossing over happens is known as chiasmata. Okay, this chiasmata can be visualized. हम देख सकते हैं, we can see under microscope during diplotin stage, because the cross which was formed like this, it was initially a closer. Now what happens? It starts separating like this. Okay, you understand the cross over here, as you can see, I am showing with my pen. So it starts separating. Then only you find that it is interacting with each other. Correct? So this what happens in diplotin. And next is diakinesis. Diakinesis is the last stage, 5th stage of meiosis 1 of prophase 1. So what happens here? Terminalization happens. So this cross which was happened, it starts moving to the terminal point, end point. Correct? This is known as diakinesis stage. So we have covered 5 stages. Now after prophase 1, what happens? We have these chromosomes, then we have the metaphase where these chromosomes will align themselves. But here you have to see that we have the homologous chromosomes. We do not have here, we have the homologous chromosome. You see, homologous pairs. Now in metaphase they are arranged at the equatorial position. Let's see if you have some questions. Sir, if we have cell performing only karyokinesis, how does that affect in equational division? Okay, so I actually couldn't get your question properly, because you are asking if a cell is just performing karyokinesis. Okay, karyokinesis means the nucleus will divide. That means, in fact, these, if I got your question right, these are certain anomalies which can happen, which you will talk about when we are talking about diseases. So here the nucleus can divide, but at times it is not followed by cytoplasmic division. So you understand what will happen? A single cell will have 2 nucleus and its chromosome number will double. That is 46 into 2 in case of human beings. So these are aberrations. Sometimes we have conditions like XXY. So we have extra X chromosome because the cytoplasmic division did not happen. So I'm really happy that you are thinking these type of questions, right? So I hope you are understanding. That's the reason these questions are coming up. So let's see. If you have another question. Okay, synaptonemal complex. Okay, yeah. So very good. I actually just skipped this word, synaptonemal complex. So I'll just see. Okay, fine. So synaptonemal complex is actually, this is the portion you are seeing, right? So here what happens, this bivalent wire, I have said you, here what happens, the bivalent, they come together, correct? They come together. That is one paternal chromosome, one maternal chromosome, they come together, crossing over happens, and this together is called the synaptonemal complex. Okay? Now let's move on. We have talked about diakinesis. So we were heading towards a metaphase. In metaphase, what will happen? They have aligned at the equatorial position, and in the next phase, that is, let's draw it here. In the anaphase, what will happen? These will get separated. Now, can you tell me what is the difference between anaphase of this meiosis 1 and anaphase of mitosis? The difference is that here the homologous chromosome is separated and not the chromatids. So initially we had seen that this chromatid was divided, right? But here the chromosome is there. That is, one paternal chromosome goes and maternal chromosome goes over here. And similarly it is followed by what? After anaphase we have telophase. So it will again form nuclear membrane and slowly and steadily Okay, so which phase, I've got a question, Edward, which phase is best for counting chromosomes? Okay, so Edward, just can you remind what I said about when we can visualize the chromosome? It is the metaphase, right? So a dividing cell in metaphase is the best phase to count the chromosomes. And actually this is a good question, because often we get this question in your examinations. Okay, so let's carry on. We have this again in the telophase. What happens? Similar to the phases which we have already discussed in mitosis. So telophase, it forms the nuclear membrane, and what will form, the important factor will decide is, we have two cells, but here we have nucleus in which we have one of the homologous pair. Unlike the mitosis, we had just a sister chromatid, but here we have one of the homologous pair. Okay, now we have considered meiosis 1. Now let's move on to, quickly we'll move on to the next part, that is meiosis 2. Okay, meiosis 2. Now I'll tell you that between meiosis 1 and meiosis 2 there is a very short phase which is a resting phase. Okay, so this phase is not very mandatory. This phase may happen in certain cells and may not happen in certain cells. This is not mandatory, but very short resting phase. This meiosis 2, I'll just tell you and explain you in one word. Meiosis 2 is equivalent to mitosis. Okay, this is equivalent to mitosis. Here the two cells we had initially. So here we had the, so I'm just drawing this cell. Inside we have the nucleus. Here we have this blue, which is, I said, the paternal chromosome, and here we have the maternal chromosome. Now can you tell me, each of these cells, if it undergoes a division which is similar to mitosis, what will happen? I'm sure you must have guessed it. It will form, this will form two cells, this will form two cells, correct? What will be the condition of the cells? Here we have one part of this, one sister chromatid. Here we have the next sister chromatid. From here we have, so let's do it with pink. Let's suppose this is pink. So here we have this one sister chromatid, and also here we have the other sister chromatid, correct? So we have 4 chromosomes, 4 cells, sorry, it's not chromosomes, it's 4 cells which are haploid in number. So I got a question from Deepankar, in which phase bacteria occurs? So I didn't get your question, which phase bacteria occurs. If you talk about that, so bacteria actually is a prokaryote, right? So I'll talk about it very shortly, because you have asked this question. Bacteria performs different modes of cell division. It can form binary fission. Remember the way I said that animal cells is pinching off? It can actually pinch off in from one cell to 2 cells. It can do conjugation, which is a type of sexual reproduction, not exactly sexual reproduction. Okay, it can do reproduction with the help of F plasmids. Okay, F factor is known, it is known as F factor. It can do asexual and sexual reproduction both. So basically, if I talk about, it can do reproduction with the help of binary fission, that is just simple division. Okay, division into 2 cells, and we need not bother about the nucleus because there is no nuclear membrane bound nucleus. So we were talking about this meiosis 2. After meiosis 2 we have 4 cells. Now what will be the characteristic? These are haploid. Very important, you should understand that these are haploid. Unlike in mitosis, were they haploid? They were not haploid. They actually had the same type. It was equational type of division. But here it is not equational type of division. It actually has 2 phases. First we have seen reductional division. Why? Because the chromosome number became halved, n to n to n, because here we have n number of chromosomes, not 2n, right? Now it has become haploid because we are diploid organisms. We have 2 copies, right? Now here we have the haploid, and these are actually gametes. You understand? These are the gametes. Now think that one of the gamete, it reacts with the other gamete of mother. It will result into the fertilization process, which will give a diploid, that is 2n number. Now you understand why the process of gamete formation happens, so that after the process of fertilization we have a diploid chromosome and the number of chromosome is maintained. You think if this reductional part, which is the reductional part? That is meiosis 1. अगर meiosis 1 नहीं हो रहा, अगर reductional part नहीं होता, तो हर बार जब भी सेल डिवाइड कर रहा है, every time the cell is dividing, it will double, it will increase. That is from 46, it will become next 46 multiplied by 2, correct? Because it is not half, it is not halved. So that's the reason. I'll ask you a question. How many chromosomes sperm cell will have? Will it have 23 or will it have 46? I'm sure you can answer, you can guess now. It will have 23 number of chromosomes because it is haploid. Similarly, if we talk about the ovum or the egg, it will also have 23. That's the reason a sperm cell, when it fertilizes with the ovum, it results into a human being which is having 46 number of chromosomes. Correct? So this is all what we have talked about the type of division in cell division. And here I have said I will be talking about one extra type of division, that is amitosis. What is amitosis? This is a type of division in which the nuclear membrane does not dissolve. Understood? So these are some of the cells, like there are certain yeast cells, certain fetal cells, they perform amitosis where there is a cell. So here we have the nucleus. The nuclear membrane is there, okay. Inside this we have the chromosomes. Let's suppose the chromosomes are here, but the cell will divide. What will happen? This is the cell. The nucleus grows. Nucleus forms 2 inside the cell. But you understand, the nuclear membrane actually has not divided. How the nuclear membrane division happens? If I draw the nucleus, it grows in size. Slowly it becomes like this, and it pinches off to form 2. So here the nuclear membrane is intact, and then the cell actually divides from here and it forms 2 cells. So this is the process which is known as amitosis. How it is different from the normal cell division? Here the nuclear membrane does not dissolve. So here is what we have covered in the very important topic, cell cycle and cell division. I hope you have understood this topic, right? If you have any questions, quickly put up these questions, post it. So I'll just check if you have any type of questions that we have. Okay, so I have let's see if we have an important question. Which state of meiosis does the genetic constitution of the gametes is finally decided? Okay, so the genetic constitution will be decided after meiosis 2, correct? Why? You understand that the genetic condition will be decided because here we have the haploid number. Now this will decide that if this is interacting with this one, with another from another parent, what type of organism will be formed? What characteristics of that organism or the human being will be formed? If this one is interacting, it will form again different, because we have seen that there is crossing over happening between the various areas of the chromosome. So you understand there is 23 pairs of chromosomes. So there is possibility of having so many crossing overs. So there are various possibilities. So after actually the process of meiosis 2, we have the gametes, and after the fusion we decide what characteristics would a particular organism have. In fact, if I tell you, after the process of meiosis 1, the fate of this gametes is already decided, correct? But if you ask about the fate of the organism, that is decided after the process of meiosis 2. So again, if you have any questions, please post up. I'll be answering all these questions. So I hope you have understood all these. So please take a note on these diagrams. These are very important. So let me see if we have some questions. Why not? So why not after Anaphase 1? Okay, so you are asking why not the fate of the gene is after Anaphase 1. Okay, so yeah, so look, this is the total phase of I'm talking about. I have actually answered you about the major phases, that is meiosis 1 and meiosis 2. If you talk about that, yes, Anaphase 1 can be the answer. So yeah, so I'll tell you one thing. So this is a very tricky question. So sometimes we get these phases, like you can be asked about meiosis 1 or meiosis 2, which of the stages define the fate of that gene. of a particular cell. Options can be meiosis 1, meiosis 2, mitosis 1, or you can have options which is like metaphase, anaphase, telophase. So anaphase, yeah, it is actually the area, it is actually the stage because it is dividing, correct? So you have got it right. Anaphase can be the answer, correct? So again, please put up the questions, we can answer it quickly. So yeah, you can just put up any questions. Let's see. Okay, so there is a question about the tapetum of polyploidy, right? So polyploidy, this is a different sector, actually different area. So once someone asked actually the question, if what happens if something is not followed by the karyokinesis process. So what will happen? Without the process of karyokinesis, the nucleus will not divide, the cytoplasm will not divide. So that will result into the cell which has got two nucleus. Two nucleus means it will be polyploidy. That is, here we have seen diploid. Now, if a human cell has 3 nucleus or 3 copies of the chromosome, you understand? So I can tell you one thing. So here we have seen. That these are the chromosomes, correct? So after separation, if suppose this did not divide, this did not separate properly, then what will happen? We'll have something like this, correct? We'll have something like this. This did not divide. This did not divide. So it will result into polyploidy. That means unwanted chromosome will come over there. One chromosome can come. So I said you that there are conditions where people can have 23 plus 1. Okay, that is they will have 47 chromosomes. So these are certain diseases. There are certain diseases known as Down syndrome. Okay, so these are certain diseases where some extra chromosome is there. Some may have X0. That means the second chromosome, sex chromosome, has not been inherited. Correct. So these are certain conditions. These are abnormalities. So these may happen when incomplete cell division happens. Correct. Okay. So any other questions? Let's see. If you have some other questions, please ask it. I hope we have covered most of the topic and you have understood it. Please post your questions again. So we'll be closing it soon because we have already covered the most of the areas, right? So here I'll maybe we can just do a summarize it quickly. Okay. So before that, we'll check if we have any questions. Let's see. So before we move on, I'll just mention that I have already talked about that we are conducting this type of live sessions. I hope you have enjoyed this. So this was the first biology session. We are conducting other sessions also. Tomorrow, don't forget to catch up with us because we have a physics session over here, right? So our expert from physics will be talking about a certain topic in physics, right? We'll be talking about that. So do not forget to invite your friends and keep updating about these sessions, okay? Let's see again if you have got some questions. Yes, I've got a very interesting question is does meiosis play a role in evolution? What do you think? Yes, it is. The answer is yes. And can anyone think about it? Can anyone say me why this meiosis process, I'm telling meiosis is involved in evolution? Yes, we have talked about one stage, that is recombination. Right? Recombination, where did it happen? It happened in the chitin stage of prophase 1 of meiosis 1. So because of this recombination, we see different variations of the characters, and if these variations, these mutations, they happen because of the cell division, these variations, if they start accumulating, this results into the process of evolution. So I'm really happy you have asked this question. That means actually you have understood this topic very well. So yes, the answer is yes, evolution is done by meiosis process. So let's see if you have other question. Again, I think I'm getting a question about abnormal type of cell division, right? Okay, fine. So yes, abnormal cell division, this can happen actually in different areas or different phases. If this separation, the reductional phase does not happen, then what will happen? One of the cell will receive an extra chromosome, that is poly, right? Multiple number of chromosome will have more than one, as I have talked about. It is X plus something, or X plus Y plus something, okay? Or it can happen 47 number of chromosome, or in fact we can have the opposite one. That means we can have reduction of the chromosome. Now imagine if one of the cell has achieved or got one extra chromosome, what will happen to the other cell? It will receive one less chromosome, right? So if that cell actually carried forward, it will result in the reduction in the number of chromosomes. Okay, let's see again if you have questions. Okay, as I've got a question, what brings about cleavage of an animal cell and after telophase? Okay, yeah. So really, this question is very good question, but you need not know about this right now because it's considered in a higher standard. But I'll definitely try to explain you. So I've said that there is a form of furrow, right? So what happens? यहाँ पे जब cell divide करना है, उनको pinch off करना है, right? It has to pinch off. So what will happen? There are certain proteins. I'm not naming the proteins, but there are certain proteins which accumulates at this point. Okay, as you can see, I'll just draw it here. I think you cannot see. So they accumulate at this center point. So this accumulation happens, constantly happens. So this protein, it accumulates and keeps on increasing. So this actually forms a separation. That's the reason we are saying it as furrow. Actually, it doesn't deepen like this, but yeah, because of this protein, this protein has the characteristics to pull this cellular membrane inside. It pulls inside. That's the reason the furrow forms. Unlike what happens, so here I can point out the difference. Okay, यहाँ पे cell, animal cells के लिए surface से शुरू हो रहा है, right? It is starting from the surface. But plant cells के लिए कहाँ से हो रहा है? It starts from the midpoint. यहाँ से एक cell plate proteins. There are various proteins actually. So यहाँ से cell plate formation शुरू होता है. I hope you have understood this. So we have almost come to the end of this session. So again, welcome to Toppr. Please subscribe our channel, right? And invite your friends. I hope I have helped you out with this topic. We'll be conducting more sessions on biology, and we are also having sessions. Tomorrow we have a physics session. Invite your friends and subscribe to our channel. If you have any doubts, please keep posting. We'll surely get back to you. Enjoy and all the best. I hope you have understood this session. So keep posting your questions. We'll surely get back to you. Thank you.